Hello, my name is Jyoti Patel and I'm a member of the European Society of Cardiology's Scientists of Tomorrow. Today I'll be talking to Professor Claudia Monaco, who will give an introduction into CyTOF, time of flight mass cytometry. Claudia Monaco is a professor of cardiovascular inflammation at the Kennedy Institute of Rheumatology at the University of Oxford, and her lab investigates inflammatory cell biology in experimental models of cardiovascular disease and also in human disease samples. So Claudia, CyTOF is a relatively new technique. How did you come across it and why?
Well, we had a long-standing interest in understanding inflammation in atherosclerosis, and in particular in human vascular tissues, and understanding why cells get activated. My interest there has always been innate immunity, because these are really the initiating steps of inflammation. So when we started studying immune cells in atherosclerotic lesions, we were really surprised by their phenotype, their behavior. So they don't behave like normal inflammatory cells. They're extremely activated in a way that would be, for example, in inflammatory in tissues from patients with inflammatory diseases. So there was a need really to find a way to understand the heterogeneity of these cells. And these cells are hugely autofluorescent. So every time we try to apply flow cytometry, it was always it's quite a challenge, because you might, especially in human tissue, it's quite difficult, especially on the myeloid populations like macrophages. We were mostly interested because these are the populations of cells which bear the innate immunity receptors, the toll-like receptors we were studying. So when I came across this technique at a conference, I was really excited. This has been developed by Gary Nolan's lab, and Sean Bendel published his first paper, and it was really a step change in the field of immunology. So that's why we became very interested. We adopted very quickly. We got one of the first instruments that was available on the market, and we started troubleshooting since, trying to apply to cardiovascular disease.
And what level of understanding does one need to perform a CyTOF experiment? Is it something that anyone can learn?
Yes, it's really something anybody can learn. The most similar technique is flow cytometry, which is a well-established single-cell techniques, and everybody who's worked with inflammatory cells before would have access to that. And in mass cytometry, the only difference is that instead of using fluorochromes as tag for your antibodies, you're using elements of the periodic table, which are very heavy and rare, and they're not present in the human body. So they're perfect tags, which can discriminate cells better than fluorochrome tagged antibodies. So your experiment at the beginning is very simple. You have cells, the antibodies, so you would stain like you would stain in a normal flow cytometry experiments. And the CyTOF facilities are now in the dozens all over the world, and it will be easy then to collaborate with a center which has a CyTOF where you can actually run your samples and troubleshoot whatever you need to make it the protocol more appropriate from your tissue of choice.
What are the tools and software which might be required for a CyTOF experiment?
Again, this is an area of really high bioinformatics activities. Now there are more and more softwares. There are a lot of free tools and software which are available on the web. There are tools which are accessible through online services, for example, DVS cytobanks. So there are really quite a lot of tools, and it's not difficult to get to learn about them and apply them to your samples. We will offer some examples later when we go in the lab or later on in the podcast. But really, this is an area where I want to say, really watch this space. There are new softwares coming up every time, and a lot of them are free, and there is a lot of tutorials on how to use them, and so this is an area of fast expansion.
So how can CyTOF help us in cardiovascular research, either a clinician or a scientist in training, doing their PhD or their postdoctoral training in cardiovascular medicine?
Well, I think if you are interested in looking at cell heterogeneity in any samples, can be samples from the blood, so the liquid biopsies, can be PBMCs that you already have stored from a large or a small clinical trial, or even if you want to understand tissues, you can perform cell dissociates. So I think CyTOF is going to be really a new step in a variety of settings of cardiovascular research, where wherever you think, wherever you have an heterogeneous cell population, and you want to understand the properties of each little sort of cluster of different cells. So I think it's very important. I'll make an example, which is in the field of inflammation, for example, in atherosclerosis, and here you can see how some inflammatory cell subsets are actually detrimental, so they will really be pathogenic. Others instead are quite protective and can heal the plaque inflammation and prevent excessive tissue remodeling and rupture. So whenever you suspect the cellular heterogeneity is impacting your model and your sample, so maybe the outcome of your patients, CyTOF is really going to provide a lot of answers for your research question.
And finally, what are the challenges left in the future with CyTOF? Are there any new tools or software which will be available in the future?
Yes, I think CyTOF is really in its infancy. So it's we are only at the sort of third generation of this methodology. So there are technical advances as we speak. There have been already several advances from the first generation, which is, I just mentioned one, which is actually how many cells are actually recovered, how much information you can recover from the cells has improved dramatically in the latest generations. And there will be more advances coming. And one of the advances is coming live this year, where the first imaging CyTOF has been released by Fluidigm, and this is able to apply these metal-tagged antibodies onto tissue sections. And by using a sort of a recovery methodology very similar to laser capture, you are going to get the same information but from tissue sections, which I think is going to be a very exciting step forward for the methodology.
So thank you, Claudia, for giving us an overview of CyTOF. And now we will go to Claudia's presentation, which will go through the principles of CyTOF.
The concept also we discussed before is a concept of cellular heterogeneity. So cellular heterogeneity is important in shaping what is the tissue sort of structure, and that has an important effect on the actual outcome of the disease. What does it mean, a cellular heterogeneity? So as clinicians, we all know that sometimes with patients, in most cases with patients, one size does not fit all. All patients need tailoring of therapy, and the same can apply to cells. For example, if you were to look at a few macrophages and you will analyze them all at the same time, you would, for example, with the PCR and look at the gene expression, you will get an average of all these different cells. But that average macrophage will not tell you anything about how these macrophages are actually different from each other and how they arrive to that average. So that means that there is a significant heterogeneity within ostensibly similar populations, and we need to quantify the molecular event with the single cell resolution. So CyTOF really is really a response to that need. For example, we use with CyTOF, there is availability to use up to 100 non-biological elements. So you have really an abundance of tags that you can use. These signals are discrete. They are not overlapping. As you can see here, you can have distinct peaks of several elements. And also you will see that we'll go through that later, but no compensation is required like you would need it in flow cytometry. And there is no background in terms of autofluorescence, since these elements that we use to tag the antibodies are very rare in biological samples. So this gives you the possibility to take an unbiased samples of single cells. It could come from the blood, it could come from the tissue, and apply sort of a single cell analysis, which then will give you the possibility to cluster these different cells and perhaps find culprit cells through all the bioinformatic tools that allow you to discern this heterogeneity of cells. So when I say culprit cells, I mean, for example, you can find what are the most pathogenic cell types in your tissue of choice. So the talk will go into the beginning of a technical introduction. Then we'll look at the vascular tissue application, blood application, and also a little bit of experimental planning. So CyTOF combines the advantage of single-cell high-speed analysis with the ability to resolve over 100 metal probes with minimal signal overlap, and allows for deep phenotyping of heterogeneous cell populations with, combined with functional profiling in a single sample. So that means you can look at immunophenotype of the cells, so what the cells express on the surface, what's the signaling status, is any of the molecular determinants of signaling, are they phosphorylated? So that means it started the sort of the cascade of signaling, and which cells are producing which cytokine or chemokines. All this information you can get it in one go. And this is the workflow of CyTOF, and the ones of you who are familiar with the flow cytometry will recognize many of these steps. So cells are fixed and permeabilized, they are stained with the isotype tagged antibodies, and we use this sort of heavy metals into the periodic tables, and then cells, once they've got the antibody on, they are going through the machine that you will see later, and here cells are ionized and become basically cloud of ions. These cloud of ions are then directed into a time-of-flight chamber, and the metal ions of interest are resolved as they pass through the TOF chamber, and then the data are generated in an FCS format, which is a very similar format, same format of what you get for flow cytometry. So we spoke before about flow cytometry, and I think that's the most sort of similar techniques that exist so far. So I really want to go into the advantages and disadvantages side of flow cytometry, because these are very revealing. The advantages is that there is a minimal spectral overlap. Because you can see that the peaks are really quite discrete. Instead, for example, flow cytometry peaks, you can see each fluorochrom can invade, let's say, the detection area of the next fluorochrom, and so the peaks are not so discrete. CyTOF instead will give you that possibility, and that possibility comes with the higher dimensionalities. You can have more markers at once. And we spoke before about the theoretical limit of 100 sort of elements. However, at the moment, the elements that you can find in which are pure enough to be used for this detection is around 40 and 50. So this is really the current panels that we are using are encompassing 45 parameters, and these parameters are a combination of antibodies and other signals which will tell you about the viability of your cell, or if a cell is actually a cell. CyTOF is also quantitative, as a broad dynamic range, and as I mentioned before, there is really very minimal background. there is really there is no analogy to autofluorescence. There's very nil biological background for lanthanide metals, which are conjugated to the antibody probes, and is not light and time sensitive. But of course, there are disadvantages. CyTOF is a destructive technology. There is no way to recover cells. You cannot direct them to sorting. It's much slower. And for some tissues, there are practical limits which allow from 1,000 to 400 cells per second. And also there is a major hiccup is the cell transmission efficiency, because you may lose half of your cells that you introduce inside the CyTOF. You cannot set gates. Actually, you completely lose what is your forward and side scatter in flow cytometry. This can give you really some trouble in identify your live intact cells, especially if you are looking at tissue digests. So the data analysis, however, it's a very important element of your experiment, because as I said, you can use up to from 35 to 40 antibodies in your sample, and of course you would have quite a lot of information at the same time. And this information, of course, you can look at this information with the traditional dot plot. You look in flow cytometry, where you will have one marker and the second marker along the y and the x axis. But you might want to look more generally at the expression. And we will go into this sort of multi-parametric data analysis softwares that reduce the dimensions of your data to something a bit more manageable, and it tells you how the cells are clustering together. So one of the first tools to analyze CyTOF data has been developed by Gary Nolan's lab, and this is SPADE, the spanning tree progression analysis of density of normalized events. And this is a clustering algorithm. A clustering means putting cells which are similar to each other together. And perhaps they could share a certain number of markers on the surface, and they would be all clustered here, while others which do not share the same markers will be clustered elsewhere. This clustering algorithm is based on the downsampling of events, and this can lead to it's very good identifying low density clusters. So this is a very important concept. And this relatedness of clusters, so how cells are similar to each other, is expressed in a dendrogram. So it's showing, it's almost like a tree shape, like it says in the name. And you can overlay the expression of markers on top of that tree. And this can give you a visual idea of why the algorithm particularly put that specific set of cells onto that specific sort of branch of the tree. A second and very useful algorithm is VISNE. This was developed by Dana Pear's lab, and is based on the T-distributed stochastic neighbor embedding TSNE algorithm that you could have looked into in a single cell sequencing and other sort of more genomics type of approaches. But here it's been transformed into VISNE to give also a different visual appreciation. Again, is a dimension reducing cluster. It will take a clustering algorithm. It will allow the visualization of high dimensional single cell data into a two-dimensional scatter plot. So again, cells which are more alike stay together, and you can, so the cells' position reflect their proximity in a high dimensional space. And the expression of each molecule acquired in the experiment can be overlaid onto the VISNE map to visualize its expression. So it's a very informative algorithm. And these are the two basics of the analysis, and we will go through some of these elements of the analysis much later on. So now I wanted just to give you an idea of the vascular tissue applications of this methodology. So in cardiovascular disease, our technology of choice, it's really analysis of whole tissue with the immunohistochemistry or histology. Analysis of whole tissue has got important advantages. So one is the tissue localization, the other is the preservation of the microenvironment in which the cells are. But you can incur into the limitations that are limited markers, and also laser capture in situ hybridization can be laborious and again is reliant on single markers. So really, if we want to understand what is the cellular heterogeneity of the tissue you are looking at, then some techniques which rely on tissue dissociation might be advantageous, because you can have single cell data on the tissue digest. The data are multi-parametric, they are multi-dimensional, but of course there are some drawbacks you have to be aware of. This can affect cell viability, can affect cell phenotype. There are, for example, in cardiovascular research, you might take advantage of experimental models of disease, and we use a lot of tissue dissociation of several organs. It can be the heart, the aorta, also lungs. And you can apply on this a single cell dissociate. Usually apply flow cytometry, but we started applying CyTOF to understand the cellular heterogeneity in these cells. And you're familiar on a sort of immunophenotyping of a murine aorta. This technique is being used since quite a lot since Galkina et al. Developed the methodology. And relies, for example, on at least a 10-color panel where you can look at the heterogeneity of different cells, and it works quite well. I just want to show you how CyTOF data look when you apply mass cytometry to a murine aorta. These are studies we performed in hypercholesterolemic mice, which are deficient in lipoprotein E. And I just want to show you how it's easy to identify several cell subsets and what we call the immune landscape of this aorta by following and adopting this technology. First, I want to show you the data. The data are, if you want in a reassuring manner, this is what you would see in your flow cytometry plot. And you can still look at the data under this guise. You can identify very nice separate cell populations, can be your lymphocytes, neutrophils, macrophages, or dendritic cells. But you can also, as I spoke before about this Wisne plot, and here I just want to show you how is a Wisne plot is going to look once you are analyzing a murine aorta cell digests. And you can see how overlaying a specific marker, you can recognize major populations, for example, your B cells, your T cells, your more myeloid markers. For example, you can see these neutrophils are high in CD11B and GR1, which is a classic neutrophil marker in mouse. And you can recognize the macrophage populations here, and the dendritic cell populations. Okay, so that's how the analysis can be done. You can look at the whole aorta, so you can take all the cells that are coming out of the aorta digest. So you can take the inflammatory cells and the vascular cells together, and you can look at some markers which can guide you in understanding these clusters. So you can see how many different clusters of cells are inside the aorta. The majority of these clusters seen are CD45 positive, highlighted here, but you have also CD45 and negative cells, for example, CD31 positive cells, which are endothelial cells, or smooth muscle cells, which express smooth muscle cell actin. And of course, you can, the more antibodies you use, the more you can actually recognize these clusters in separation. So here I'm going to focus more on the inflammatory cells because that's the interest in our lab, but this technique can also be adopted on CD45 negatives, which will be the more vascular sort of cell types. So then you can then gate on your CD45 populations, you can run another Wisne analysis. And every time you run a new Wisne analysis, you will have a different looking plot because the cells are reclustered again according to the algorithm. And you can see here how you can identify the more inflammatory cell populations, even more detail. You will identify the B cell populations and neutrophils and eosinophils and lymphocytes and NK cells and other ILCs, innate lymphoid cells. Then you can take a step further if you are more interested, like we are, into the myeloid populations, and you can exclude the lymphocytes and K cells, ILCs, and you can rerun another clustering algorithm that will give you something of this shape. And I'm going to describe a bit more in details these populations. And you can see here the overlay, for example, of CD68. CD68 is widely expressed on a variety of populations in the plaques. And this, we knew that already from flow cytometry, but here you can see with even more detail how different populations are starting expressing CD68 once they get into the plaques. So CD68 is perhaps a very broadly expressed. It won't help you much with the clustering, but with the use of other, at least 15 markers, you can actually address these populations very well. You'll be able to distinguish, as I showed before, neutrophils from eosinophils that we find in the plaque. We can find the two classical DC populations. This is CDC1s and CDC2. You can find defined monocytes populations, and you can find a rather large population of macrophages which are present in the tissue. So with the flow panel, this would be not possible to distinguish necessarily all the populations at once, because probably you're relying on 10 markers and you will have definite issues in identify sort of these bona fide populations within, unless you use several different panels. But here you can do only one in one go in the same sample. So I think this is a very powerful technique to understand what's the immune landscape of the aorta, and also to phenotype what changes to a specific gene or specific environment or therapeutic intervention in experimental models will lead to changes and relationship of these populations. When we applied CyTOF for the first time, we started seeing that one of the more heterogeneous populations in the plaque, perhaps that is expected, were actually the macrophages. So we decided to focus on these macrophages, trying to identify handy cell surface markers which can help us cluster this population of cells, identify them, and perhaps in the future sort them and study their biology much more sort of in detail. So then we ran a Wisne on this macrophage population, which are CD11b positive, CD68 positive, CD64 positive. And one of the first surprises for us, it was also the healthy aorta has a significant, great deal of macrophage populations already residing into this in the murine aorta. And once we analyzed the CHO-fed ApoE mice, so these mice are hypercholesterolemic, but they're not taking a high-fat diet, and this is quite later on in the disease, we saw that in reality there were only two populations of macrophages which are quite significantly different from an healthy aorta. And this is this cluster, and you can see here, this other, when you start high-fat feeding the ApoE knockouts mice, these populations are even more enriched. And these clusters in particular are CD11C expressing macrophages, which really seem to be the newcomers onto the plaque. And we think that is a very interesting population. We have published recently in Circulation that this population is dependent on a transcription factor is called interferon regulatory factor 5. I'm not going into this data at the moment, but this is really just an example of how you can identify cell populations of interest, and by understanding what are the cell surface markers, then you have a handle that will allow you to sort this population aside, for example with the flow cytometry approaches, which allow recovery of live cells and study them further into your system. So I showed you before some examples of hypotheses that you can address with CyTOF, but maybe the concept I really want to highlight is that really there is almost no limit to the number of subsets you can actually identify by tailoring your panel and adding crucial markers. You will achieve separation and segregation of the different clusters and cell types from each other. And I think that's important. This is a sort of very field which is really starting now, and it will lead to really understanding what is the heterogeneity of the cells in the mural and aorta. Right now, I would like to give you some snapshots of human applications to human vascular disease, and this is really what this is the first piece of data that I saw on CyTOF, and that's what really got me very interested in this technology. Because we all know that when we study human diseased vascular tissue, one of the biggest enemies is actually this tissue autofluorescence that is very well known also in other imaging technology, but can really cripple analysis by flow cytometry. You can see here instead how CyTOF by not being dependent on fluorochromes, which can be interfered with autofluorescence of tissue and lipid-rich cells, you can really achieve a nice separation of populations of cells. So this is a simple example of the potential of this technology, specifically in the artery, where autofluorescence is almost is more disturbing than in any other cell types. To show you, for example, this technique can be applied to human carotid and arterectomy tissue, where you can identify populations of cells with clarity. You can identify your CD4 T cells, CD8 T cells, B cells, NKs, neutrophils, classical dendritic cells, and the various myeloid populations. And as an example, I wanted to show again how the CD11C macrophages we spoke before feature prominently in human atheroma dissociates. You can see almost a very good majority of cells which are expressing CD64 are also expressing CD11C. So this is again an important marker we carried from the mouse to the human, developing this cellular hypothesis on these cells. And similarly, we can apply to human aneurysm tissue, and here again you can identify your myeloid cells and CD4 cells and quite a lot of inflammatory cells which are present in these tissues. However, so I focused a lot on vascular tissue, but in reality CyTOF is born to analyze blood. So PBMCs and other blood cells are really a very easy target for CyTOF, and I really want to show you ways you can analyze cells and give you some example. Before we were more looking into one clustering algorithm or Visne. Here we will look more at other analytical tricks into this in PBMCs. So one of the most important things is analysis in the analyzing data is actually verifying that the cells that you are actually run through the machine are actually bona fide cells. For example, you might encounter in the blood quite an awful lot of platelets, that you can identify rather easily. And it's important that you focus on your live intact cells. We'll go through later more on what this means in an experimental setting. So as I said, now, I showed before more the algorithm Visne. He said now we're going to show in blood application more as an example of the SPADE analysis. And if you run peripheral blood PBMCs from a patient, and you run the algorithm, you will arrive to this sort of tree of different cell subsets and possibilities. So once the algorithm has run, that's there is interpretation. That's when the interpretation of the user comes in, and on the basis of the overlay and of the analysis of the dot plots, you can recognize each single branching, and you can name each single branching on the basis of current knowledge. So here, basically, you can see how by using CyTOF and by using a SPADE algorithm, you can really recognize different subsets of cells in the blood with the incredible detail and certainty. You can also use this technology to look at intracellular cytokine staining. For example, you can use stimulated and unstimulated conditions, and you can see, for example, how an important cytokine, tumor necrosis factor alpha, is activated is produced by monocytes after LPS stimulation. Right, now I want to run through some elements of experimental planning. So time in a CyTOF study really divided into, like in every experiment, into three components. So one is experimental design and reagent validation, experimentation, and data analysis. What is different with CyTOF is actually that most of your time will be spent in experimental design and reagent validation and the data analysis. So this requires, this preparation phase is going to be extremely long compared to the actual run of the samples, where this is the easiest part of the experiment because you have validated all your antibodies, you are acquiring samples which can be extremely quick, your workflow is extremely quick, you just need to add the antibodies to the cells and you run an experiment like a flow cytometry experiment. So it's the beginning. And then the data analysis, then it will take you months and months. So you can run one experiment perhaps in one day, or even a fraction of the day, depending how many samples. And then the data analysis might take you 3 months, just trying to understand what are the specific of that results of that population. So what are the differences between patient groups. So I wanted to spend some time in this experimental design and reagent validation phase. So it's really important, like in every experiment, to define what is the biological question. And because it's a complex technique, you want to know what is the perfect experiment, but what also could be the compromise experiments. And what is important in this validation phase is important to have cell lines which are expressing the markers you want to study, find out what are the commercial sources of antibodies against the surface proteins, and find out what are the best clones to detect this surface or intracellular targets in your hands. One note of caution is that the sensitivity on lanthanide tagged antibodies is lower than the most popular fluorescence reporter. This is because the chelating polymers that are used to plant these elements onto the antibody allow only 100 metal reported ions on antibody molecule. So it's, this can prevent analysis of molecule with really low signal to noise ratio. So it's very important to know that your molecule, you can detect it by flow cytometry. If you can detect it by flow cytometry, CyTOF will be very easy. But otherwise, if you don't detect any signal with the flow cytometry, CyTOF is perhaps not the best technology to move into. Another important part of the experiment is really the antibody validation and titration. So the antibody panels must be validated on live and fixed cells to optimize the surface marker staining. And you have to see and study in very much detail how this antibody staining is affected by fixation and permeabilization protocols, and even by the barcoding of the samples. And the titration is particularly important, even more than in flow cytometry. And one of these reasons is that you don't want excess antibody, which will have still quite a lot of elements that can impact and reduce the half-life of the detector. So you cannot stain, like in flow cytometry, in excess of the target. You really need to have enough signal to be detected. So titration of an antibody is really important in this experiment, and it will be important to perform in-house antibody conjugation and titration. Sometimes these levels of antibodies that you use might be different from tissue to tissue and preparation. So it's quite important to study in the lab these aspects. And then, again, this is just to give you an overview of the CyTOF workflow. We spoke about the antibody titration. You can buy off-the-shelf antibodies, or you can conjugate the antibodies yourself. There are kits for conjugation. We spoke also about the flow cytometry comparisons and what are advantages and disadvantages. Now it's important to know that your sample is being run through flow cytometry to verify its expression ability to be detected. And we spoke also about the need to optimize the protocol to your tissue. Another aspect that it's important, specifically in the setting of clinical trials and multi-center sort of application, it's very important to, there are very specific sample preparation steps, some of which we will discuss later when we go in the lab. And it's important to verify that your PBMCs and conserved cells that you already have stashed are preserved in a way to allow enough cells to be run through the CyTOF and enough cells to detect the subsets that you want to detect. And of course, we spoke about the tissue digest and that you use for tissue analysis, and how it's quite important to have it tailored to this technology. Of course, the rest of the experiment is very much into sort of will be in the charge of the CyTOF central facility to which you're going to run your sample with, and really that's they are going to look into the daily maintenance of the instrument, the tuning, the weekly cleaning, and look and perform all the quality control for the instrument. Data analysis can be performed with a variety of software and bioinformatic tools. We are looking only at some. I showed you only two of the algorithms, but now there is a proliferation of algorithms that are being developed. This is really an exciting field where there will be more and more tools available for clustering these cells and recognizing their differences. So I just want to touch that one of the future developments, which is actually already here, and it has been recently released, the imaging CyTOF, which will allow the detection of and the analysis of tissues on slides, and it's a combination of laser capture and sort of a CyTOF. And so this has been recently published, and we are really keen on to trying it on very soon. Thank you for your attention, and now we're going in the lab just to look at one of the CyTOF experiments of today, where Jenny is waiting for us.
Hi, I'm Jenny Cole, a postdoc in Claudia Monaco's lab, and here I've got my single-cell suspension of aortic cells, which we've already prepared. The cells here, we have sort of anywhere between 100 to sort of 500,000 cells per aorta. For CyTOF, most things have been optimized for staining 3 million cells at a time. And so for this reason, we generally pool aortas from different mice to have enough cells for one sample. It is possible in CyTOF staining to barcode your samples and run them and stain them all together. For this would allow you, if you're, say, collecting patient samples, would be to collect them all at once, keep them frozen, and then when you come to do the staining, you defrost them all, and you can then barcode them and stain them together, and then after the experiment and the cells have been captured, you can then debarcode them and separate them back out into individual samples again, which can help with staining consistency and variability. And the first thing we need to do is to wash the cells a couple of times in metal-free reagents, just because the CyTOF is very sensitive to all metal contamination. So we have special metal-free buffers that we've purchased for this purchase. So first of all, I'm just going to wash the cells. So now that we've washed the cells, the next step is to add a rhodium intercalator. And we do this to be able to discriminate the live and the dead cells from each other, as the rhodium intercalator at this point will only go into the cells that have got compromised cell membrane. So this then goes in the incubator for 15 minutes. Okay, so now we've done the rhodium staining and we've washed the cells a couple of times. And now we're going to block the cells with FC block just to prevent any unwanted staining. So we're just going to incubate this for 10 minutes at room temperature. And at the moment the cells are in 50 microliters, and the next step is going to be adding the antibodies. We should do a 1:2 dilution, so they'll also be in 50 microliters. Okay, so now we're going to add the antibodies. And the antibodies for CyTOF are conjugated with various different metals. At the moment we can do a panel of up to 35 different antibodies against either surface or intracellular antigens. Not all molecules that you might be interested in, there may not be a commercially available antibody that's CyTOF ready. In this case, if there's an antibody against your antigen of interest, you can conjugate the antibodies yourself with metals, and that's what we've done for some of our antigens of interest. Today we're staining a fairly general panel of surface and intracellular markers of different types of leukocytes. So the antibodies have now been added to the cells, and we're going to incubate those for about half an hour at room temperature. If we were continuing to do intracellular staining, after this point we would fix the cells, permeabilize them, and add the intracellular antibodies as well, again for about half an hour. So we've now stained with the antibodies, and we've washed the cells twice in cell staining buffer. If you've done intracellular staining, the cells are fixed. At this point you have to increase the spin speed to make sure that you don't lose too many cells. So now we're sort of reaching the end of the protocol, and we're going to add the second intercalator, which is an iridium intercalator. And this is in a fix and perm buffer. And what it does is it goes into every single cell, and it will allow the CyTOF machine to actually detect the cells. And so this then can either be incubated at room temperature for about an hour, or it can also incubate in the fridge overnight or up to sort of 48 hours. So now we've finished the iridium stain, so we now have to wash the cells 4 times, twice in the cell staining buffer to remove any excess iridium, and then twice again in water, because the samples need to be in water to be run on the CyTOF machine. In the final water wash, we're going to count the cells, and we're going to then adjust the concentration to be around 500,000 cells per ml. For an aorta, they optimally run at around 0.3 to 0.5 million cells per ml, but for other cell types, this concentration is different and needs to be titrated on the CyTOF machine.
So we're now at the Botany Research Centre at the CyTOF facility. My name's David Ahern and I'm the facility manager. And I'm going to introduce our Helios CyTOF system. I'm going to explain briefly how it works, and then we're going to acquire Jenny's sample that she prepared this morning. So I'm going to turn the machine on. It's going to get quite a lot noisier in here. So basically it's very similar to flow cytometry, but instead of using fluorochromes to conjugate our antibodies to, we use metal isotopes. And basically the way the machine is set up at the moment, we have our sample here in the sample introduction system, and the whole point of this front end that you can see is to take our sample of cells which are in water and to exchange the water carrier for argon and to introduce the cells in a single one at a time basically into the system. And to do that, basically at this point we've got a heater which evaporates off the water and replaces the carrier solution with argon. And right in here we have the actual plasma, which is where the cells as they come through get vaporized and ionized, and at that point each cell will become a cloud of ions essentially, and that cloud of ions will consist also of the ions that come from the metal isotopes connected to your antibodies. So that ion cloud then goes into the time of flight chamber, and basically what happens in here is that each ion takes a certain amount of time to get through the chamber and reach the detector, and that is dependent on the mass of the ion. So that way the detector can tell which ions are hitting the detector at each time, and that relates to the quantity of the antibody that's bound to the cell originally that has gone through the machine. And then basically the computer takes that information for each cell and outputs the data as an FCS file, which is essentially the same file that you get from a flow cytometer. So we can use that file in any analysis packages that we use for flow cytometry. And yeah, so that's basically how it works in a nutshell. Okay, so now we have Jenny's sample that she's finished preparing. So it has been washed in cell buffer twice and then washed in water twice. And this is now ready to acquire. So basically we're going to put it on the sample loader. And the sample is now ready to acquire. So we're ready to acquire Jenny's sample on the CyTOF. So basically what we're looking at now is a preview screen. And along the top here, what we've got is all the channels that we have in the current panel that we are measuring. And these include a lot of Jenny's staining panel, which is this area here. And right at the end, we have the iridium stain that she did, the final stain that she did in her prep. And in the middle here, we have also some background channels that we monitor. So basically what's happening at the moment is we run the cardiovascular samples quite slow through the machine. It helps with the resolution of the cells, is what we found. But what you can see is these kind of blobs that are appearing on this live update. And what these are is basically the cells going through the machine and hitting the detector. And what we have is the signal being detected in various different channels as it goes through. So what you'll see very bright is the DNA intercalator stains here for the iridium popping up, along with some other markers within the panel. So this is one cell, for example. This is another one and another one. So once we have it set up in the preview mode, we can then just hit record and start acquiring the sample for real. And we can set it to acquire either to reach a certain number of events or to record for a certain period of time, whichever suits the experiment. Okay. So what we can also do is look at a live update of what cells have already been acquired on the machine. So this is what we've got here. And what we're looking at is the DNA intercalator stain, so the iridium on the bottom, and CD11B expression on this axis here. So these are basically our CD11B positive cells as they're going through, and this updates as it goes through. So it helps us to see that everything is working okay. And we basically just let it run until it reaches the end of the acquisition time or the number of cells that we wish to acquire.
So now we've acquired our cells on the CyTOF, we can now move to the analysis stage. So before we actually do any analysis, we need to normalise the samples. And we do this because over the course of running during a day, the CyTOF machine tends to lose a little bit of sensitivity. If we're running lots of events, say with an aorta, they may take a couple of hours to run on the machine, and therefore over that time there could be variation. It also means that if you're doing stainings and analysis and acquisitions on different days, they can all be normalised together and analysed together, and you've got more confidence in the consistency of the data. And to be able to do the normalisation, every sample has been spiked with the calibration beads, which have set metals on that we know where they should be. So there's different softwares that you can use to analyse your CyTOF data, including FlowJo, R, but the one that we most commonly use is CytoBank. So this is CytoBank. When you go in and create a new experiment, you give the experiment a name, who the researchers are and the purpose of the experiment, and then you can add your files. So the files that come from the CyTOF machine are FCS files, and you just drag and drop them or select them in here, and they're uploaded into CytoBank. So this is my APOE aorta, and that's uploaded. And then we go into the working illustration. And here you can start doing various things, and you can analyse your data in terms of whether you want different conditions or time points. But the first thing that you do would be to gate the cells. So into the gating, this is how it initially will look, plotting event length against time. And the first thing we would do is plot the iridium intercalator against each other, which is cells 2 against cells 1. And it's this bright population at the top here that are probably our single cells. We can then take those single cells and look for the rhodium intercalator that we stained for that would have labelled our dead cells. And you can see on the gate drawn here, that's the live cells that are negative for the rhodium. We can then take these live aortic cells and we can look for CD45. The aorta has a lot of non-CD45 cells, such as endothelial cells and smooth muscle cells, and there will also be debris. But for this experiment, we're interested in the CD45 positive cells, which you can see here. Once these are gated, you can then gate for any other populations that you're interested in. Here we have CD3 for T cells on the x-axis, and CD19 for B cells on the y-axis, showing both these types of cells are present in the sample. Here we've gated CD64 on the y-axis against CD68 on the x-axis, which shows a macrophage population. So once you've performed all the gates you want to do, there's further analysis that you can do, which will include clustering of the samples and looking at populations. So the one we commonly use is VISNE, so we'd create a new VISNE analysis and give it a name. And then we select which population of cells we want to use. So here we want to use the ones I've gated as being CD45 positive. If you had multiple samples at this point, the software can take the event count, and if it does proportional sampling, it will take the same number of events from each sample and put them for analysis. For VISNE, you then need to choose which channels you want to cluster on. So which markers that you stained for do you think will identify the populations in your sample. So for these, I'll have selected most of the main ones I expect to find on the leukocyte populations. And then you can click Run VISNE. And this will then take a while to run, anywhere between sort of 15 minutes and a few hours, depending on how complicated you've made the analysis and how many cells that you're analysing. So now the VISNE is completed, you can see that we can go back into gating within the VISNE experiment, and we now have two new channels in the analysis, this TSNE2 and TSNE1, and this is just the way that it's displaying the VISNE. And you can see now we've got islands of different islands which may be different cell types. We can then change to have a colouring channel which will colour these events based on the expression of a various marker. So if I change the colouring channel here to CD19, which would highlight B cell populations, you can see that this island down the bottom is coloured red, And this is a heat, basically a heat map with the highest expression will be displayed in red and the least in blue. So this population at the bottom are most likely to be a population of B cells, which they can then be gated. And then changing the colouring channel to something like Ly6GC or GR1, this would highlight a population of neutrophils. I can then change it to, say, CD68, and so this highlights the monocyte macrophage cells within the sample. And for this, you can gate all the different populations, you can look through all your different markers and see the expression of whichever antigens that you've stained for. And you can go on and do further analysis of these subsets, and you can also use the other tools within Cytobank to also do further analyses.
So hopefully this podcast has given you an overview of CyTOF, its applications in cardiovascular science, and this will benefit your further research. Thank you.